Hello everyone, welcome back to Python for Bioinformatics. In the previous lesson, we learned how to check whether a pattern exists in a DNA sequence using the in operator. But often we need more than a yes or no answer. And today we will learn how to find the exact position where a pattern occurs using Python's find method. Now suppose we have this DNA sequence and we are interested in the pattern CGTA. Knowing that the pattern exists is useful, but in many biological situations, we also need to know where it occurs. For example, we may be looking for a motif, a primer binding region, or a specific sequence feature. Before thinking about Python, let's think about the problem itself. If I asked you where CGTA starts in this sequence, you would probably scan from left to right until you find the first occurrence. And that is exactly the task we want Python to perform for us. Python provides a string method called find. The general syntax is sequence.findPattern. We give Python the pattern that we are searching for, and Python returns the starting position where the pattern begins. And if the pattern cannot be found, Python returns -1. Let's try our first example. We have our DNA sequence stored in the variable named as sequence. The important part is sequence.find CGTA. Here, sequence is the string being searched, find is the method, and CGTA is the pattern we are looking for. When Python runs this code, it scans through the sequence from left to right and looks for the first place where all 4 letters appear together in the correct order. And Python returns 3. This means that the pattern starts at index 3. Remember that indexing starts from 0. So biologically, we can say the motif CGTA begins at position 3 in the sequence. Now looking at the index map, the result becomes easier to understand. We can see that the first C of CGTA is located at index 3, and which is why Python returned 3. Now let's search for a pattern that does not exist. The code is identical except the pattern is now TTTT. Python again scans the sequence from left to right, and this time no match is found, and therefore the output is -1. -1 is a special value used by the find method to indicate that the pattern does not occur anywhere in the sequence. In biological terms, the motif we are searching for is absent in our sequence. Now here's another important behavior. The pattern AT appears multiple times in our sequence. Now what do you think Python will return? And Python returns 0. That is because find always returns the first occurrence, and as soon as Python finds a valid match, it stops searching and reports that position. Now let's summarize the meaning of the possible outputs. A positive number means the pattern was found and the number tells us where it starts. 0 means it starts right at the beginning of the sequence. And a value of -1 means the pattern was not found. Now time for a quick practice question. We have a new DNA sequence and we want to find the pattern AAC. Pause the video and try to predict what Python will return before looking at the answer. Now let's check the solution. Python searches for AAC in this sequence and returns 6. Looking at the sequence and index positions, we can see that the pattern begins at index 6. And this confirms that find returns the starting position of the first occurrence of the pattern. So today we learned how to locate patterns inside a DNA sequence using the find method. The syntax is sequence.findPattern. The method returns the starting index of the first match and returns -1 when the pattern cannot be found. This makes it a very useful tool for locating motifs and sequence features. Thank you for watching. In the next lesson, we'll learn about the count method, which helps us determine how many times a nucleotide or pattern occurs in a sequence. See you in the next video.